Well, hello everyone. So I'm Joseph Holden, and I'm one of the Freshwater Quality Research Programme champions here at the University of Leeds, along with Pippa Chapman, who we'll hear from a little bit later on. So welcome to the February 2026 programme webinar. The programme is actually now being extended and will run through to 2028. And consists now of 6 projects. So we have the champion team based out of the University of Leeds, and now we have a new project which is going to be starting in March 2026, looking at the river systems, catchment systems around Lough Neagh in Northern Ireland. So we'll, as soon as we're able, organise a presentation from that project team, the ClearNEA team, to kind of update you on what that project's going to be doing and the type of work or research that activity within it. And then we have our sort of ongoing other projects. There's 5 other projects, which includes the Quantum project, which is looking at nutrient enrichment and ecological impacts of livestock farming. Pacific, which is looking at the ecological impacts of different chemicals, and particularly kind of some of the microbial contributions. LTLSFE, which is a large-scale modelling project, which is going to produce some amazing forecasts and model outputs across the whole of the UK. And then EcoMix, which is a project looking at mixtures of chemicals and how that's impacting freshwater biodiversity, and particularly it's been looking at the whole suite of the Yorkshire river catchments. But today we're going to hear from the MOT4Rivers project, and our webinar series does involve this mixture of projects which are within the programme, but then presentations from people from outside the programme, and then some international presentations as well. So we're trying to kind of mix that up. This is a little poster that was created by one of our artist teams, James Mackay, and of course this was representing the programme with the 5 projects, but of course we've now got the Lough Neagh edition. But the principles are still the same in terms of we're trying to transition from, you know, what's bad on the right to what's good on the left, basically through what policy interventions, what practice interventions, what public interventions we can make to improve the quality of our freshwaters. So we're hoping the programme will be really influential and impactful. So we're looking forward to hearing the kind of questions and discussion later on from those of you who are joining live, and that's the benefit of joining live, that you can partake in that discussion, and thinking about what the implications are of some of the research work that's been done for delivering this improvement in UK freshwater quality. So do please get in touch if you've got any other ideas for how the programme can connect with you or your organisation through the email or the website or the other information that's on screen there as well. So just do engage with us. Okay, so I'm delighted then to hand over to the MOT4Rivers team, who are going to present today, and we're really looking forward to hearing an update from their project. So over to you all. I think, Miriam, are you kicking off?
Yes, thank you, Matt. We'll share the screen, and there'll be 3 of us presenting. Okay. So yes, good afternoon, and thank you very much for the opportunity to present the progress of our project here. 3 of us, myself, Matt and Anna, will be representing a large team that's behind this work in the MOT4Rivers project, and we'll be specifically updating on the progress of work package 3 that's focused on the modelling of multiple pollutants, and in this case pharmaceuticals in particular. Next slide, please, Matt. So just a very brief recap. The MOT4Rivers research project set out to explore this concept of ecological safe space, to understand the safe space within which ecosystems can thrive. And this is delimited by changing hydrological, but also chemical and biological stresses. So looking at two axes here, looking at future hydrological extremes, whether they are low flows or high flows and more regular extreme high flow events, or the chemical stresses. So our aim is to build this understanding across scales, from catchment to national scale, to understand the impact of hydroclimatic controls on the fate and impact of pollutant cocktails, to examine the sensitivity of ecological communities and the structures and function to multiple stresses, and finally to deliver a decision support tool for testing interventions for mitigation and adaptation. So in this talk today, we'll be particularly focusing on the third objective, to look at how far we have got with developing a decision support tool that looks at the risk from pharmaceutical pollution and in combination with hydrological extremes. Next slide, please. So just a little bit of background, why pharmaceuticals? Chemical pollution is one of the 9 planetary boundaries that have already been exceeded, and human pharmaceuticals are increasingly detected globally across freshwater systems. We need to therefore mitigate the ecological and human health risks arising from this pollution that may have impact on behavior, reproduction, and health of wildlife, and also, as we know, is thought to lead to increasing occurrence of antimicrobial resistance in the environment. Pharmaceutical fate is controlled by processes that are uncertain and variable both in time and space. And this includes both prescribing rates, so the consumption of pharmaceuticals, human excretion, removal during wastewater treatment, and also the hydrological dilution capacity in the freshwater systems. Therefore, we need decision support tools that can integrate these diverse processes, but also explicitly represent uncertainty, because in many places we have very uncertain data or even lack of data to characterize these processes. So here I'll hand over to Mads to describe how we approach this problem.
Yeah, as Miriam was saying, it's what we are going to present today is sort of well, it's kind of 2 components. There's one component that's been looking at doing some hydrological modeling to understand current and future risk to pollution from extreme high and low flow events, and then the main part of the talk is about this probabilistic modeling of pharmaceutical pollution risk from sewage treatment works that we've been developing using a Bayesian network approach, and how we've applied this model to the River Amund catchment with the specific aims of simulating spatial and temporal concentration patterns and risk of 16 pharmaceuticals under sort of current or recent historic conditions, and then try and compare the simulated concentration against field to infer key processes controlling risks, and also explore the potential impacts of management interventions, climate change on pollution risk using this tool. And finally, we'll show some initial results of how we've tried to upscale this risk model to sort of more national scale application. Right, so the pharmaceutical model we've developed is conceptually very simple. So it's just based on a simple mass balance. So the model, it considers a catchment upstream of a monitoring point along the river. We then look at how much pharmaceutical mass is being prescribed within the catchment, how much of this is being excreted after consumption, and therefore ends up in wastewater, how much is being removed during wastewater removal, and therefore how much remains and ends up in the river. And to calculate the concentration in the river, we simply just divide the total upstream pharmaceutical load from the wastewater treatment plant with the river flow. And finally, to evaluate the ecological risk, we then compare these predicted concentrations to predicted no effect concentration thresholds. So very simple, and this is an approach that have been used before, but where we've done things a bit differently is that we've implemented this into a Bayesian network. And why have we done that? Well, as Miriam mentioned, the main reason is that all the inputs required for this mass balance calculations are very uncertain and variable in time and space. And the Bayesian network approach, it provides a transparent way to sort of account for this uncertainty explicitly and propagate it through the mass balance. And importantly, this also means that all the model outputs are also described by probability distribution. So we get a probability distribution of our predicted concentration, and the risk therefore becomes the probability of the concentration exceeding the no effect concentration, which is more meaningful than just looking at a single deterministic estimate. Another advantage worth highlighting about the Bayesian network is that it allows for sort of very efficient evaluation of both predictive and diagnostic inference scenarios. So that means it allows you to both look at going from observed causes to look at what are the likely effects going to be, but also go against the errors, if you like, and go for observed effects to see what are the likely causes. And these kind of features are very useful for risk analysis and for modeling what-if scenarios. So we've applied this model to the River Almen catchment, which is located here in the central belt of Scotland, just west of Edinburgh. Within this catchment, there are 13 sewage treatment works that are owned by Scottish Water, which are shown here with red circles, and the size of each circle shows just how large they are in terms of treatment capacity, where this one here is the East Calter wastewater treatment plant, which is by far the biggest one in the catchment. Within the catchment, 2-year monitoring campaign has been carried out where pharmaceutical have been measured monthly at 20 different locations in the catchment, which are shown here with the blue dots. And we've been monitoring for 16 pharmaceuticals. And we can also see, I've plotted the sub-catchment for each of these monitoring points as well, based on which you can sort of see that some of the monitoring points are sort of upstream them. There are lots of sewage treatment work discharges, whereas others, they have no sewage discharges. So to apply the model, we first need to derive all the input distributions. So in order to derive the input distribution for flow, we used hydrological modeling to simulate the flow at the 20 monitoring locations because, well, all the locations are engaged, so they had to be estimated. And for this, we used SWOT, which is a widely used hydrological model. And the model setup and calibration was done by University of Edinburgh, and specifically by Anna, who's going to talk a bit later on this later. So based on this model, we could derive continuous flow time series for each of the monitoring points. And we could then summarize these on a monthly basis for each location, which is what this plot over here shows. The model was also used to simulate future flows under climate change. So for this, the climate change projections from UK CP18 and Eurocodex were both used, and in both cases we used the RCP 8.5, which is sort of considered the worst case scenario, and this allowed us to simulate, I think it was 54 sort of ensembles, members of what the future flow might look like up till 2080 at each of the locations. For the prescribing data, we derived these from Public Health Scotland. So we got monthly prescription data from what is it called, the Prescribing Information System database, which contains prescription data from all GPs and the community pharmacies and from hospitals and so on across Scotland. And we then summarize these on a monthly scale as well, and spatially we summarize them on a health board level. And this was then used as input for the model. Finally, we were looking at the excretion. So excretion rates were compiled from literature, and it included over 300 sources. And the result of the reviews, what we tried to show here in the figure to the left, and it just shows that there's a huge variation between, or the excretion of the different compounds varies substantially between the different compounds, but we can also see there's some compounds where we have a lot more data to base this on than others, and that the variation for some compounds is much larger than others. Similarly, we did a review literature of the removal rates during wastewater treatment, and that's what's shown here to the right. So we also supplemented this by estimating removal rates from paired wastewater influent and effluent data in SIP2, the Chemical Investigation Programme. So in total, we got removal rates from over 200 wastewater treatment works, and we tried to group these into different water treatment categories, as shown along the axis here. So we have secondary activated sludge, secondary biological filtering, and then tertiary activated sludge and tertiary biological treatment. And again, we can just see there's just a lot of uncertainty in the removal rates, and a lot of variation going on. So we now got all the inputs we need, and once we feed that into the model, what we get is a probability of the predicted concentrations at each of our monitoring points, and we then compare this to the observation data that we got. And to sort of evaluate the model performance more quantitatively, we calculated so a distant measure called Wasserstein. So it's not so important how you do that exactly, but just note that smaller distances mean that the observed and simulated concentration are quite similar, whereas larger distances means sort of poor agreement between the two. And any value less than 0.3 corresponds to about a twofold difference, whereas a value over 1 means over an order of magnitude difference. So this heat map just shows the result of this. So we have the different pharmaceutical compounds along here, and we have our monitoring location down here. So what we can see here is that there's certain compounds where we seem to be doing pretty well across most locations. So as for example, trimethoprim, tramadol, and sulfamethoxyl. I can't remember how you say that exactly. And there's also locations where we seem to be doing better than other locations. So particularly monitoring point 1 and 8, perhaps the locations where we do the best, but also monitoring point 11 looks pretty decent. And if we just show how the distribution look like for these two monitoring locations and for these three compounds, we can see it looks pretty convincing actually, with the Well, considering we've done no model calibration whatsoever, I think, yeah, it's quite a convincing result. If we look spatially where we are in the catchment, we can see that monitoring point 1 and 8 are in the main stem of the River Amund and the two locations that actually received the most treated wastewater. So it's maybe not surprising that these are the location that performs the best, because it's kind of these locations where the assumptions behind the model are most valid. Maybe more interestingly, it is to look at where the model does not perform so well. Again, we can see there's certain compounds where we do quite poorly. So there's a plot here of sulfadiazine, ibuprofen, and ranitidine. And again, this is for monitoring point 1 and 8. And we can just see that for ibuprofen and sulfadiazine, we appear to underestimate the concentrations, and for ranitidine, we seem to overestimate. This is perhaps not so surprising, given that sulfadiazine is widely used as a veterinary medicine, and we know ibuprofen has a large over-the-counter consumption, which is a source term we've not included or accounted for in the model. Ranitidine was banned from use in 2020, so it's quite likely that we've overestimated the source term in our model here. Similarly, we can start looking at locations where we seem to do quite poorly as well. So monitoring point 2, 4, and 17, there's a lot of red in these columns here. And here's just a plot of naproxen and ibuprofen. And again, we can see the model underestimates the concentration quite significantly. We can then look in the again where these points are in the catchment, and if we highlight the sub-catchment for monitoring point 4 and monitoring 17, well, first of all, there's only very small wastewater treatment works, more like community septic tanks located in the upstream of these points. But if we plot where there are CSO outfalls in these catchment, we can see there's quite a high density of these within the catchment. So it is quite likely that this could explain the concentration difference here. Similar, we can look at monitoring point 2, which is highlighted here, the catchment of this. And we can again see there's some CSO outfalls in this catchment, but this here shows a plot of the private septic tanks that have been that are located within the catchment. And again, we can see there's a very high density of private septic tanks, which again could possibly explain why we observe much higher concentration than we simulate with the model, because this is a source term that's not included in the model at the moment. Finally, we can also look at monitoring points that have no sewage treatment works upstream, and where our model, because it only accounts for sewage treatment work, basically simulates zero concentration. So we can see at this monitoring point 19 that this is the concentration of the different pharmaceuticals that have been measured. At monitoring point 19, we can see there's quite high concentrations, and that clearly indicates presence of unaccounted sources, which in this case is quite unlikely to come from CSOs. At least there's hardly any known CSO outfalls within the catchment, whereas there's a lot of private septic tanks in the catchment. And finally, it's possible that diffuse sources could also contribute. Right, I'll just pass on to Anna next. She'll talk about the hydrological modeling results. Thanks.
Thank you, Mads. So yeah, as Mads commented, the University of Edinburgh was the one producing the hydrological model. And apart from doing this, we included the climate change projections, UKCP 18 and Eurocordex, and we also did some analysis to analyze the future floods and the droughts over time. These analyses were, well, the climate change projections were from 1990 to year 2080. Next, please. And what we saw from the relative changes and the absolute changes over time in floods is that in the downstream area of the catchment, there is a higher variability and higher accumulation of flows, which can translate in future risks in flooding and contamination. Next, please. What we observe for the droughts over time is that in this case, in the middle catchment, in the middle area of the catchment, the variability is higher, together with higher periods of with drier periods, and this can be understood with a potential risk of environmental stress in that area. Next, please. So we also wanted to include some parameters that allow us to analyze the risk of flooding events over time, and we consider the risks of pharmaceutical pollution from CSOs, the pesticide pollution, increased flow, improving river quality, and land runoff events. Next, please. And what I'm showing you in here is a quick example of the risk of pharmaceutical pollution from the CSOs over time. Here again, I'm showing you the absolute changes and the relative changes, and what we can see over time is that the risk of pharmaceutical pollution it's higher. Well, it's high everywhere, but especially in the downstream area of the catchment, and there is a high variability as well. Next, please. And just here to summarize, this is the big picture of what we can expect for the future in the catchment. So going from low risk to high risk, we can see that in the upstream region corresponding to sampling station 12, we can we're experimenting a smaller flows in the future, but this is more showing less risk. This will be more sensitive to proportional changes, so making these stations early indicators of the system as a wide stress system. Sorry. Then moving to the midstream area with showing a medium risk corresponding with stations 8 and 11, we can think that this might serve as reservoirs for accumulating contaminants. This is probably a risk of delayed release in the future. And finally, if we observe the downstream area of the catchment corresponding to station 1, we observe that there is a potential of this area to become more vulnerable to overflushing and to pollutant loading. And well, we want to know if these the same locations that are also risky from a pharmaceutical pollution viewpoint. Then I pass this to Miriam.
Thanks, Anna. So basically, we tried to bring these two pieces of work together, the hydrological risk analysis and also using our probabilistic model to look at pharmaceutical pollution. And we have done this for the 16 pharmaceuticals that were mentioned before. On the left-hand side, you can see evaluation of risk, so exceedance of what's deemed to be predicted no effect concentration from an ecological point of view. So the red would be the probability of exceeding these predicted no effect concentrations. And each panel here shows one of the monitoring points in our study catchment, and each bar within a panel shows 1 month from January to December. And here we show an example for azithromycin, that's one of the more risky compounds based on our analysis. And what we can see here is that the risk clearly increases in the summer due to reduced dilution. So that is driven by the hydrology and the future change in hydrology as well that Anna has referred to. Again, we see increased risk at the main stem of the river, as was the case in the hydrological analysis, so monitoring point 1, monitoring point 8, for example, and monitoring point 11. But we also found some risk in the tributaries, and that may relate then to reduced dilution capacity of these tributaries. And when we compare the current situation and the future, we can see a lot more red in the future in our bars here in the bar chart, and also we see higher risk as represented here by the paler colors on the main stem of the river, but also in some of the tributaries. So clearly we can expect climate change in particular to have an effect on increased risk in the future. Next slide, please. So we did look at some potential mitigation scenarios that would be relevant in this case for sewage treatment works. So we looked at the effect of theoretical reduction in prescribing rates, bearing in mind that the population is actually predicted to age. So we would need to consider how realistic it is to expect reduced prescribing. But nevertheless, as a thought experiment, if you like, or one of the potential measures we should be looking at, moving towards green prescribing rather than using pharmaceuticals so much to address various health conditions. We did look at the effects of reducing prescribing rates by 10% and by 50%. And the effect of this was proportional on the risk reduction. So for 10% reduction, you can see a great change in the risk levels. So the current situation is in black, and the 10% reduction would be in yellow. And then for 50% reduction, you do see already a marked reduction in risk. This is for clarithromycin, diclofenac, and ibuprofen at two of our most risky locations on the main stem of the river. We also looked at how the risk would be affected if the treatment, given the current technology, was, let's say, optimized at each treatment works. So we applied the 90th percentile of what we know about current removal rates. So if every treatment works was working at the close to highest possible level of what we know about their current capability, how would the risk be reduced? And that is termed optimal treatment here in green. And we can see that it does seem to have quite an effect on the current situation, which is in black. But it did vary with the type of treatment works that are in place and also with the compound. Because obviously for compounds where we can already remove them well, we don't see such a great improvement. For compounds where the removal rates are more variable currently, the improvement would be obviously much better. And then we can combine these scenarios. We can think about optimizing the treatment to optimize removal rates and also reduce the load that enters the sewage treatment work through changes in prescribing. That's a maximum scenario, and that's the red line here. And that does show that we are managing to affect the risk levels quite markedly. Next slide, please. What we then did in the final stages of this work, we upscaled the model to 40 camel catchments in Scotland, using the same assumptions and the same data, but obviously made specific for each of the catchments. And here I'm showing examples of two of the compounds that show the highest risk. The good news is that the other compounds show lower risk, and in the case of those compounds, the risk does tend to be mostly focused in areas with highest population density, so in the central belt of Scotland. But to show the gradation here and the variability, I've picked azithromycin and ibuprofen. And in the top panel, we can see the situation in January, so when we have the highest dilution in our streams, and the bottom panel shows the situation in summer when the dilution is the lowest. And you can see that clearly there is a huge effect of hydrology here, and the risk is much greater in the summer due to reduced flows. And the risk does tend to be greatest where population is obviously most dense, but we do observe risk in this case also in the northeast and northeast of Scotland in other locations. And the next slide, please. So in conclusion, we have so far developed this part of our model, the sewage treatment works, and we can see that they are a significant contributor in many places to the pollution. But we have also identified that other sources are likely to contribute in some areas. So in future work, we are planning to develop additional modules for this model where we will represent the losses from septic tanks, combined storm overflows, and diffuse sources. We have found hydrology as an important driver and influencing the risk. That's apparent in the current conditions, but also under future scenarios. And we can see that significant improvements in removal efficiency or reduction in prescribing may be needed for the highest risk compounds to reduce the risk levels. And our final slide just shows the full team that contributes to the MOT4Rivers project. If you can just bring that up, Mads. And we would like to thank our funders and acknowledge all the universities that are participating in our project. Thank you.
Thanks so much.